For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. Online Molecular Biology. Assignment Help. Question 1. You are interested in understanding the residues of a specific group I intron that function specifically in catalysis of the splicing reaction. To probe this question, you mutagenize the intron and, using an in vivo screen, isolate mutant variants that are splicing defective. Because group I introns contain many regions of secondary structure that are important for folding of the RNA, but probably are not directly involved in catalysis, you decide to initially examine your mutant RNAs for those defective in folding. These mutants will be set aside, as they are unlikely to have specific defects in the catalytic steps. 1. You find that it is relatively easy to isolate the unprocessed RNA from your mutant cell lines. With this in mind, suggest a type of analysis you could use to probe for mutants that have defects in RNA folding. Explain what controls you will use, and how you will decide if the mutant is a folding defect. You can identify mutants in RNA folding by isolating the unprocessed RNA from WT and each of your mutants, and labeling it, digesting each with either S1 nuclease or snake venom RNase individually and running the products out on a denaturing gel. S1 nuclease will digest ssRNA and SV will digest dsRNA, who will compare the banding pattern in each of the mutants to the wild-type pattern. Any footprints that differ from the WT will indicate a mutant in RNA folding. Nursingassignmenthelp.com. 1b. After setting aside the folding mutants, you wish to further screen your mutants for those that are good candidates for having specific catalytic defects. You decide it will be easier to look for mutants that have defects in the second splicing step. Your idea is to look for mutant RNAs that accumulate the intermediates expected of an RNA that can catalyze the first step successfully, but show a complete defect in the second step. Given the schematic of the full-length pre-RNA shown below, draw the structure of the RNAs you would expect to see after an in vitro splicing reaction for a mutant RNA that is defective in the second step. Also, in addition to your purified pre-mRNA, what components would you add to the in vitro splicing reaction? Nursingassignmenthelp.com. 1. Guanine nucleoside or nucleotide 2. Divalent cations, e.g. Mg++ or Mn++, 3. Monovalent cations, e.g. Na+ or K+. 1c. Using the strategy outlined above, you successfully identify several mutant RNAs that are specifically defective in carrying out the second splicing reaction. You are most interested in mutants that identify residues that are truly part of the active site that catalyzes this reaction. Therefore, you next want to determine if the mutant RNAs are or are not defective in the tertiary structure rearrangement that is necessary prior to the second splicing step, as mutants defective in this rearrangement reaction are less interesting to you. Suggest a genetic or biochemical method that will allow you to screen out mutants that are defective in the rearrangement. A general description of the method, without experimental details, is sufficient. What result would expect for the rearrangement defective mutants? You would like to eliminate mutants that are defective in tertiary structure formation. Normally, regions P1 and P7 come together in step 1 and regions P1 and P9 come together in step 2 of group I intron splicing. Nursingassignmenthelp.com. These interactions can be tested by using a cross-linking reagent, 3 points. Since you would like to identify mutants in the active site of the RNA, you will study mutants that cross-link similarly to the wild-type RNA, but fail to catalyze the second step, 2 points. Question 2. You are studying the pathogenic fungus C. albicans, and when you sequence one of its mitochondrial genes you find that it has a novel intron. Analysis of its sequence suggests that it may be related to the group 2 self-splicing introns. Your first step in studying the intron structure is to mutagenize it. 2a. You identify a series of mutants which can no longer splice. Shown below is the sequence of the region of the intron, not the whole intron, containing these mutations. NursingAssignmentHelp.com. The mutations in the mutant strains may disrupt the secondary structure elements necessary for proper folding of the intron into an active catalytic structure. Inspection of the sequence above reveals several potential stem loop structures in which the mutations are located. 2b. You decide to further analyze these mutant introns by treating fragments containing this region with a set of RNA nucleases. You use an RNA fragment corresponding to the intron sequence shown above, labeled at its 5-foot end, as a substrate. You visualize the digested products by autoradiography. The smallest piece you can distinguish on the gel is 5 bases in length, based on control size standards. NursingAssignmentHelp.com. You find that mutants B and C give similar patterns to mutant A, while mutants E and F give similar patterns to mutant D. From these data, draw the following. I. The secondary structure of the wild-type intron 2. The secondary structure of the MUT. A-like mutant introns. 3. The secondary structure of the MUT. D-like mutant introns indicate on each drawing regions 1 to 8. Also indicate on the wild-type structure where the individual mutations AF map. NursingAssignmentHelp.com. 2c. You decide to generate new mutations which suppress the mutant phenotypes and restore normal splicing. You isolate 2 different suppressors, identified as SUP1 and SUP2. SUP1 specifically suppresses only MUT-A, and SUP2 specifically suppresses only MUT-E. Each suppressor turns out to map to a location within the intron, but neither is a revertant. Why are these suppressing mutations so specific? Can you guess what the specific mutations might be, and where they might be located? NursingAssignmentHelp.com. The suppressing mutations are specific because they are individual base changes complementary to the original mutations and restore Watson-Crick base pairing between the 2 bases in question. This allows reformation of the original secondary structure, although with a different sequence. This is known as covariation. Muta has a U instead of a G at the second position in region 8, in the first stem structure. SUP1 must therefore have an A rather than a C at the second to last position in region 6. MUT-E changes a C to an A at the second position of region 2, in the second stem. SUP2 is therefore likely a change from AG to AU at the second to last position in region 4. 2d. If you repeated the nuclease protection experiments with both S1 nuclease and snake venom shown above using similarly labeled intron fragments from the 2 suppressed strains, one of which carries the SUP1 and MUT-A mutations, while the other carries the SUP2 and MUT-E mutations, what would you predict the digestion patterns to be? Explain your reasoning. The 2 suppressed strains are likely to contain introns which generate digestion patterns with S1 and snake venom nucleases that are identical to the wild-type digestion patterns, since WT intron splicing activity has also been restored. NursingAssignmentHelp.com.